In this video, we aim to demonstrate two methods of generating intracerebral hemorrhage in mice. We will describe a double injection model of autologous whole blood, as well as an intrastriatal injection of bacterial collagenase. Both models are commonly used in hemorrhagic stroke research. Before showing the surgery, we would like to illustrate the procedure, starting with the double injection model of autologous whole blood. This schematic represents a coronal brain section 0.2 mm anterior of bregma. The lateral ventricle is marked LV. CPU stands for putamen, a part of the corpus striatum, and GP identifies the globus pallidus. Both the corpus striatum as well as the globus pallidus belong to a group of subcortical nuclei known as the basal ganglia. A 26-gauge Hamilton syringe is positioned 0.2 mm anterior and 2 mm lateral to bregma. The needle is inserted through a cranial burr hole and advanced 3 mm below the dura. Following the insertion, 5 microliters of autologous whole blood, previously collected from the central tail artery, are injected intraparenchymally at a rate of 2 microliters per minute. The needle is then advanced 0.7 mm to the target position of 3.7 mm in depth. After a waiting period of 5 minutes, 25 microliters of autologous blood are injected into the striatum. The needle is left in place for an additional 10 minutes before being slowly withdrawn at a rate of 1 mm per minute. Coagulated blood of the first injection prevents back leakage along the needle track. In contrast, the collagenase injection model consists of a single intrastriatal injection of 0.075 units of bacterial collagenase dissolved in a half microliter of saline. Collagenase is injected at a rate of 2 microliters per minute. The needle is slowly withdrawn 10 minutes after the completed injection. Bacterial collagenase disrupts the basal lamina of cerebral vessels, causing